Hello biologists. Today we're going to learn about enzymes and some of the cool things that they do. You will be following along and recording your notes as you watch this video, so feel free to pause, rewatch as needed. Let's get started. So enzymes' main cool thing that they do is to speed up chemical reactions. And there can be two types of chemical reactions. We will learn more about these if you go on to take chemistry, but we can classify these chemical reactions as endothermic or exothermic. So go ahead and take a moment to write down this information. And let's talk about what this means. So some reactions require energy to happen. So in this first case, energy is required. It has to go into the system, like this root endo is referring to, means inside of. So energy has to be put in order for the chemical reaction to happen. And then other chemical reactions actually release energy, and we call those exothermic because the energy is exiting the system or it's being produced as a result. So we take a look at where energy is going, where it's required or produced, and we can classify chemical reactions in this way. So we know that enzymes are super important. You have many enzymes in your body helping you stay alive. Their main function right here, speed up chemical reactions, and they do this by reducing the amount of energy needed for the reaction to take place. And we're going to take a look at the next slide what that energy is. It's called activation energy, and they manipulate the activation energy so that the chemical reaction will occur at a faster rate. So let's take a look. In the first graph A, we have a scenario where these reactants are lacking an enzyme. There are no enzymes present to help them turn from reactants into products. And if we look at this graph, the y-axis is showing us the amount of energy needed, and the reactants have to overcome a lot of that energy. This hump that these guys have to travel over is very high. We call that the activation energy that's required. So what enzymes do is they decrease this hump right here, the activation energy, and that is seen on the next slide. So if you think about these reactants as like a ball or like this as a roller coaster track, they have to climb up, 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 all the way up here in order for them to have a chance to be turned into products. But enzymes come in and they are able to help these reactants become products more readily. So it's easier to get over that energy requirement. And so we see that this hump is smaller because of enzymes present in the system. So here's a review. Proteins are made up of amino acids. Amino acids will link together. They form peptide bonds with each other, and this peptide strand will fold onto itself and create some kind of specific three-dimensional structure. And that three-dimensional structure is actually very important and dictates how well the enzyme can do its job. So remember that these enzymes are made of proteins, most enzymes, and proteins are made of amino acids. So they're a specific type of protein, and they determine, their shape determines their function. So if their shape is messed up, their efficiency is also not so good. So their shape is everything. We're going to talk about how that helps them do their job. Another property is that they are reusable. So like the shoes you put on your feet today, we put them on again and again and again, and we use them over and over and over. Enzymes are just like that, in that they can execute hundreds of thousands of chemical reactions over and over and over. Eventually, they do have a lifespan. So just like any part of your body, over time things become a little worn out, a little less efficient, and eventually they will be targeted for recycling in your body, and they will be replaced by new enzymes. But they are reusable, and that's a property that we should be familiar with. So another property is that they are very specific. They are not a jack of all trades. They are a master at one job, and that one job is very specific. So you have many different enzymes in your body that help you do all the different chemical reactions needed to stay alive. And the enzyme that does a particular job is defined by its shape, and we can say its shape is everything. So we'll take a look at some images where the enzyme fits with its substrate, the molecule that it acts on, like a puzzle piece. And let's take a look at that on the next slide. So here we have, let's take a look at the image first, and then we'll talk about what this example is. So we have an enzyme shown here in yellow, right? This is the enzyme molecule called LOLOC. And then we have substrates. So in this case, we have two substrates. A and B are molecules that are going to come in and fit into this enzyme. And they're going to interact with the enzyme at its active site. So the active site is the portion of the enzyme that interacts with the substrate. So the active site shape, or the portion of that enzyme, is super important that it fits in together. Here we go. That it matches the shape, because if the substrates don't lock in there perfectly, the enzyme isn't going to be able to do the chemistry it's designed to do. So some enzymes, like this one in this image, take molecules and, like matchmakers or Cupid, they bring two separate things together, right? So these substrates are being linked together, forming some bonds, and now we have a product that is AB, right? So in this scenario, we can have enzymes bring in molecules together to form a larger product, like dehydration synthesis. Or we could do the opposite of that and break them down into smaller products, like hydrolysis. So our analogy here is like gossipers, breaking up a couple. Don't do that. That is mean. Do not gossip. But let's take a look at our substrate. For our analogy, this was one molecule, and we are breaking it down into smaller products, right? So we're breaking bonds and actually releasing the energy that was stored in those bonds in this case. So enzymes can put things together or break them down, and we don't use those terms in science. We use these terms down here. So see if you can take a moment and figure out which one. This top one is called a catabolic reaction. And we have an anabolic reaction down below. So in one case, we are taking a substrate and breaking it down. In the other one, we are taking molecules and putting them together. So see if you can match which one matches with each of our analogies, the matchmaker versus the gossiper. Okay. Another property that is important to be aware of in this class is that enzymes can get denatured. We talked previously about how their shape really dictates and defines their job and how efficient they are at their job. There are certain conditions that can denature an enzyme. So enzymes are often happiest and most efficient when they're in their preferred environmental conditions. So that is somewhere in your body with a certain temperature range, pH, and so on. So these enzymes that are found, if you think about the enzymes in your saliva, they're used to that temperature and that pH, and they're happy in those conditions. But when those enzymes move down into your stomach when you swallow your saliva with your food, eventually those enzymes are going to find an environment that they're not so happy with. They're not designed to work well in wide ranges of environmental conditions. So when we change environmental conditions like pH or temperature or something else, they get a little messed up, and we call their change in shape denatured. So if we take a look at this enzyme, it's being heated, and its active site we can see matches well with the substrate it works on. But if we heat this enzyme, the shape of the enzyme gets messed up. We call this a denatured enzyme. And if it's denatured completely, it cannot do any chemistry. So we see that the substrate just leaves the enzyme not changed at all. Enzymes do have a tolerance, so we can see that some enzymes can still function, just not as well if they are partially denatured. But if that active site specifically is becoming denatured significantly, that enzyme will not work. And finally, another thing we should talk about is that enzymes can be inhibited. So if you think about the enzyme and how it works like a lock, and it works on the key that fits into it very specifically, if something were to block where the enzyme interacts with the substrate, we could prevent the enzyme from doing its job, which in some cases is actually beneficial for an individual's health. So sometimes we have enzymes like these listed here that are doing chemistry at a rate that produces some kind of condition or disease. And if we can prevent these enzymes from doing their job with an inhibitor, then we can treat these conditions. So if you have heard of gout, gout is the buildup of uric acid crystals in joints. Very painful, I would assume. Doesn't look fun at all. But if we can target the enzyme that is building up this uric acid product, then we can prevent or treat gout with this medicine, this drug that we designed to be an inhibitor. And so what this drug does is, let's go back to our pictures here. Let's say this is the enzyme that does work, that produces the condition gout and builds up that uric acid. If we come up with a drug that could fit in and block the active site right here, then we can render this enzyme not functional. As long as we have this drug present in the system, the enzyme could bind the drug and not the substrate and not produce the uric acid in this case. So these inhibitors are things that we have designed to treat certain conditions, some forms of cancer, high blood pressure. So sometimes inhibiting an enzyme can actually be beneficial for an individual. So it's pretty cool stuff. So I hope the notes went well. If you have any questions, feel free to reach out and talk to me during advocacy, or feel free to re-watch certain clips of this lecture if helpful. So good job. Hope you guys have a good day, and I'll talk to you soon. Bye.